and control theory. I did my PhD a year ago in the EC department on data-driven modeling of dynamical systems. And in my postdoc, I have been carrying that background into study of cell dynamics. And today I'm going to present our work on multi-network RNA velocity, where we're considering both the gene regulatory network inside the cell and the cell-to-cell network to get a full picture of the cellular dynamics. One issue with the single-cell genomic is that our data set are static snapshots. At the time of the measurement, we take a snapshot of the cell state, but we have been always struggling to infer the dynamics of the cell. To tackle this challenge, the concept of RNA velocity was introduced by Lemano in 2018. The idea is that to use single-cell genomic data to infer the direction of the gene expression. Before I introduce the RNA velocity, I will briefly recap the gene expression process. So starting from DNA, it is being transcribed into nascent RNA as the transcription rate alpha. And those nascent RNA are then splicing at rate beta, where their introns are being chopped off. With this, spliced RNA are then degraded at a rate, degradation rate gamma. If we can measure the unspliced mRNA together with the spliced one, then we can get a peak of where the gene expression dynamics is going. For example, if we have a ton of the unspliced mRNA, but not so much of the spliced ones, then we are about to see an increase in the gene expression profile, and vice versa. So the whole idea can be captured into a linear ordinary differential equation. So U here is the spliced mRNA count. It is being produced at the transcription rate alpha and then spliced at the splicing rate beta. The S here is the spliced mRNA. It is being generated at the rate beta and degraded at the rate gamma. And the RNA velocity is then defined as the change of the spliced mRNA. So let's first take a look at the top right figure here. I took this picture from Lemano's 2018 Nature paper. So they used a differentiated neuron cells. The blue one here are the stem cells, and the brown cells here are the differentiated cells. So using the RNA velocity, we can not only tell where we are, but we can also put arrows on those cells, which tells us where the cells are developing into. So those RNA velocity are those arrows. It is starting from the current cell state, and the arrow are pointed towards the predicted cell state transition. In the second figure is from the Burgess 2020 paper, where they used pancreas data. We can also see that from the precursor cell state, it is then developed, differentiated, and committed to one of the 4 final cell fates. So by using the RNA velocity, we can answer the questions such as if we started in these precursor cells, where the cells are likely to develop it into, and how are those delta cells be likely generated. Currently, perhaps the 2 most well-known models for estimate RNA velocity are the original model called VeloCyto and the subsequent model called single-cell Velo. In the original model, Lemano assumes a steady-state model where the steady-state ratio of the unspliced to spliced RNA are given by the degradation rate divided by the splicing rate. The RNA velocity are then estimated as the deviation from the steady state. One of the limitations is that they assume a steady state model. Virgins then extend the steady state model to consider to accommodate a transient dynamic by considering this full dynamical model. So this full dynamic model is basically an analytic solution of those ODE we have seen on the previous slide. But still, they treat each gene independently and does not consider any cell-to-cell or gene regulatory networks. Perhaps the most related to our work is the TF value. They're considering a transcription factor, how it influenced the RNA velocity. But as we can see from their formula, that they assume a specific behavior. That is, they assume a sign function. Also, they cannot directly model the transcription factor is influenced on the transcription rate. For applications such as drug intervention, the drug will directly influence the regulatory genes, which influence the transcription rate. So ideally, we want to explicitly model how the gene regulatory network influences the transcription process. So in our model, we considered a two-level network. Inside the cell, we consider there is a gene regulatory network, so we added a term in orange here. For the gene regulatory network, we model it using a weighted directed graph, where each node on the graph is a gene, and the weight is being captured by these W matrices here. So kappa here is just a constant matrix, and the W plus or W minus are two non-negative matrices to capture the positive and negative regulatory effect of the gene regulatory network. I will explain these terms in details in the next slide. So the second level network we considered is a cell-to-cell network. So we suppose that cells interact with each other via some chemical signals, and we added a term in green here. This term essentially is a consensus term that can be justified by the spatial transcriptomic data. So let's assume the cell G have a higher level of spliced rate. Then what this term do is, since SG is higher than SI, this term will drive the network. This term becomes positive, and it will drive the network to a consensus. So our goal is to study how this networked RNA velocity behaves and how can we use it to analyze the targeted drug intervention. In this talk, I will just focus on the first one, the one regarding the gene regulatory network, and in our full manuscript, we'll consider this two-level network. To further explain how we incorporate the gene regulatory network, let's consider this nonlinear function denoted by Rg. So we incorporated this Rg and let it multiply by the transcription rate. So in this formula, this transcription rate alpha of gene G is a basal transcription rate. And this nonlinear function incorporates how the gene regulatory network influences the transcription process of gene G. So let's look at this nonlinear function Rg closely. Now, as introduced before, we use a weighted directed graph to capture the gene regulatory network. But at the same time, we still want this whole state to remain positive because the count of unspliced and the spliced one can only be a non-negative value. So instead of directly considering a directed weighted graph that can take both positive and negative values, we partition them into two parts. So the W plus are non-negative matrices that capture the positive regulations, and W minus is still a non-negative matrices, but it captures the negative regulations in the gene regulatory network. So to understand how our function RG reflects the positive and the negative feedbacks in the gene regulatory network, let's consider a simple example. Suppose we have two configurations of the gene expression, S and S hat. In these two configurations, the individual expression level are the same except for gene Q. So we would like to know the response of RG to the changes of GQ from SQ to SQ hat. So we can compute the incremental gain of RG due to such a change. And this incremental gain can tell us whether this gene Q have a positive regulation on G or a negative regulation on gene G. From this expression for the incremental gain, we can see that the regulation on gene G is positive if the W plus matrix is positive, and the regulation effect is negative if this W minus matrix, which captured the negative regulation, take a value greater than 0. So in summary, our model can account for the gene regulatory network, and the transcription process are now influenced by two parts. The first part is the basal transcription rate, and the second part is the gene regulatory network and its influence on this basal transcription rate. I will now present our first analytic result on this networked dynamical systems. So the first, we would like to know whether a steady state of this network dynamics exists. As we can see from our networked model, when we considering the gene regulatory network, now we have a nonlinear ODE rather than a linear one. And for this nonlinear system, we say there is a steady state if there is a solution by setting the right-hand side of the ODE to 0. I will not bother too much about the proof, but what our theorem says is if we set this kappa to be 1, what the theorem says is that the degradation rate of gene G have to dominate the whole transcription rate and the whole net positive regulations on gene G. Now suppose we are at the steady state and we wanted to know if we perturb the system a little bit, will the system go back to the nominal steady state? Let's first considering a very simple scenario, that is there is only one gene. So in this single gene, we go back to our formal linear ordinary differential equations. And for this linear ODE, we say it is asymptotically stable if all the real part of the eigenvalue is less than 0. So for this single gene case, we can just solve for the eigenvalues of this matrix A. And what this tell us is the eigenvalue of this matrix is minus beta and minus gamma. And the beta is the splicing rate, degradation is the gamma is the degradation rate. So when these two rates are both positive, then the eigenvalues of this matrix A is negative. So we can say in this single gene case, the system is always stable. Next, we're considering a scenario where there are only promoter in the gene regulatory network. So for a promoter, what the promoter does is it is a positive feedback for the network. And we would imagine that these positive feedbacks, if we perturb it a little bit, it doesn't have too much robustness. And in fact, the perturbation is going to be amplified because the gain is positive. So in this case, the promoter only network, the dynamics is still linear, and we can still solve for the eigenvalues of this matrix A. And because of the regulations in the network, now we have a much stronger sufficient condition for the network to have a stable steady state. And what these sufficient conditions say is for this promoter-only network dynamics, the steady state is stable if the degradation dominates the splicing rate, and the splicing rate have also to dominate the transcription rate and the total positive regulations in the network. So our ultimate goal is to investigate the stability of this gene regulatory network, where we consider both positive and negative regulatory effect. As we see, this is a nonlinear ODE. And for these nonlinear systems, we have to use tools from control theory, namely the Lyapunov analysis. So what the Lyapunov analysis do is we wanted to find an energy-like function, what is called the Lyapunov function. So if we have a function that is like an energy that is positive except for at the steady state, and if this function always decrease along the trajectory, then we can say the trajectories of the systems will always converge to this steady state, and we call this system is asymptotically stable. So for example, on the figure here, suppose we have a simple Lyapunov function, and the red lines here are the trajectory of the system. Since this Lyapunov function exists and this trajectory always decreasing along this function always decrease along the trajectory, and it's finally converged to this steady state. So in this case, the steady state is stable. By leveraging the Lyapunov analysis, we have the ultimate result for the stability of the networked RNA velocity. So the first condition here says the total negative regulation effect on the network have to be lower bounded by some delta where it's a constant, and that is scaled by the total splicing count in the network. So to explain this condition, recall that the inhibitors or the negative regulations in the gene regulatory network can be thinking of as a negative feedback. This negative feedback is going to add robustness to the whole network dynamics. So one would imagine that if there are enough negative regulations in the network, we will tend to have more stability in this dynamics. The second condition basically mimicked the one we have seen before, so it captured interaction between the splicing rate and degradation rate, which also depends on some constant depending on the network, the gene regulatory network. So when these two conditions is satisfied, we can say that the gene regulatory network dynamics is asymptotically stable. Now that we have seen the network RNA velocity, we further extend the dynamics to consider drug intervention. So this drug intervention is in collaboration with AbbVie. And the idea here is suppose we have a set of targeted gene that we want to steer the set of targeted gene to some certain gene expression level. So the question is, how could we design the drug intervention to steer the gene expression to this targeted level as fast as possible? So to incorporate this drug intervention, we're thinking of drug intervention as a control signal, and we denoted the drug intervention targeted at gene Q by ZQ. So this is a control input, and we multiply this control input to the influence of gene Q in the gene regulatory network. So in this way, we still have a basal transcription rate alpha of gene G, but in the gene regulatory network part, the influence of gene Q is now multiplied by the control signal Z. So for this networked dynamics, it is now depends on the control signal. And since our goal is to find the fastest possible intervention strategy to steer these network dynamics to a targeted profile, we can formulate it as a time optimal control problem. So the objective here is we want to find the minimum time to steer this to the final targeted gene expression level, subjected to the controlled dynamics. And the U here is some admissible control. I will not bother you too much with the optimal control theory, but I will just briefly say that using the Pontryagin maximum principle, we know that if the control signal takes the value between 0 and 1, where 0 corresponding to no drug intervention and 1 corresponding to the drug intervention is at the maximum strength, then this optimal control strategy or the optimal intervention strategy is a bonbon controller. It means at any time the controller takes the value either 1 or 0. So in summary, the optimal control theory will tell us a structure of the drug intervention strategy. Finally, I will present two simple simulations to demonstrate the drug intervention. And for the drug intervention problem, we wanted to know after we manipulate a certain gene, how the other gene in the network, the dynamics, how the dynamics of other gene in the network will change. So in the first example, we consider a gene regulatory network consists of 3 genes. The gene 2 have a positive regulatory effect on gene 3, or it have a negative regulatory effect on gene 1. So we will keep the gene regulatory network topology fixed, but we will set the transcription. We first set the transcription rate of gene 2 to some non-negative level so that the transcription process of gene 2 is on. And then at time 2, we will shut the transcription process of gene 2 off to mimic the effect of drug intervention. And we can see from the right panel that after the gene 2 is shut off, the unsplice count decreases at an exponential rate. And the splice count of the gene 2 is also decreasing because there's no unspliced being produced. In addition, we noticed that since gene 2 acts as an activator on gene 3, after we shut down gene 2, this unspliced count of gene 3 also decreases a little bit because there is no promoters for gene 3 now. And similarly, since gene 2 have a negative regulation effect on gene 1, once we shut it off, the transcription of gene 1 will actually increase. So from this simple simulation, what we can know is that after the drug intervention, not only the steady state will change to a new steady state, but the transient dynamics is also changing too. In the second example, we're considering a gene regulatory network consists of 5 genes. So now we are looking at gene 1, which have a positive effect on gene 5 and negative effect on gene 3. Like to the previous case, at the beginning of the process, the whole transcription process is on. And then at time 2, we shut down gene 1 and observe what happens to the steady state and also the transient dynamics. So compare these two panels, we can see after we shut down gene 1, the unspliced count of 1 are also decreased at an exponential rate. And gene 5, since its positive regulatory effects are removed, gene 5 also the expression level of gene 5 also decreases. And since the negative regulations on gene 3 has been removed, the expression level of gene 3 increases. So in summary, I only talking about the gene regulatory effect in this talk, but we have more in the full manuscript where we considering a cell-to-cell interactions, where we added a consensus term, and we also analyze how this consensus term is going to affect the steady state and the stability of steady state, and whether the gene expression level among the cell will reach a consensus or not. And we also examine how the drug intervention may affect the safety liability genes and how to design the drug intervention as an optimal control problems. Thank you.
Thank you for your great talk. So I have a question on, so because you use puncture eigen maximum principles to got this would be the band control. I'm really curious about, so if you multiply some scalars into the systems, like in front of the WGQ plus, that turns. So I'm curious about, so because a lot of drugs have the half-lives. So if you consider the drugs where that's scalars will have some half-lives, so if still the optimal control problem, so this would become some singular control, because if the band control, it makes like, I'm not really sure, so what's the biological interpretations of this control?
The biological interpretation of the optimal control strategy?
Yes.
So the controller is the drug.
Yes.
And the drug, we wanted to know how to design the drug intervention such that we can get the targeted gene expression level as fast as possible.
The ZQT will remain as a constant in the whole times, or this would like take the exponential decays as the time going?
Well, your question is, will ZQ be a constant?
Yes, or it will.
We wanted to solve for the ZQ. So ZQ is the drug intervention, and we wanted to know what this optimal controller will look like. So in order to solve this optimal controller, we formulated as an optimal control strategy. So before you solve this optimal control problem, you do not know what ZQ looks like. And you solve for it using it, and this optimal control strategy will tell you the structure of this ZQ. It's not a constant, so I said it's a bound controller, means it's switching between 0 and 1, but it's at which point it will switch. It's actually there is a formula for it. I didn't show you here. So it's also a solution like using the Pontryagin maximum principle. So it's not a constant.
Okay, we have time for maybe one more question.
Thank you for your great talk. I had a question about parameter estimation. So my understanding of the existing SE-Velo and VeloCyto, they look at the moments of the distribution in order to try to estimate parameters. Whereas in the final form of your equation, you also consider neighboring adjacent cells, right? And so I'm wondering, in this framework, would you still be using the moments of like the 30 nearest neighbors? And then how do you reconcile that with looking at your next nearest neighbor for the SJ-SI term?
So first, there's no learning part in my model, it's the future work. So, but I think the moment, you mentioned that the S-Velo using moment, you can actually solve the full dynamics. Or you can just group this term as some unknown parameters and directly learn that instead of learn the moment.